Bismillahirrahmanirrahim. I'm Dr. Shagufta Perveen, Department of Botany, Government College University, Faisalabad. The topic of my presentation is protective role of manganese, proline, and rice straw extract in wheat under drought stress. Water, which is one of the essential factors for the sustainability of life of all living organisms, including plants, it has been reported that annually 55 million people worldwide are affected by water deficit stress. Water deficit stress can lead to about 50 to 70% crop yield reduction globally. It has been estimated that by 2030, about 700 million people, that is about 40% of the global population, will be at risk due to water stress. Wheat production is directly associated with food security. Drought stress is a pivotal constraint to global agriculture production, especially in warm, arid, and semi-arid regions. What are different effects of drought stress on plants? Drought stress affects plants at morphological, physiological, and biochemical level. What are different morphological changes? Drought stress cause a decrease in the number of leaves, decrease leaf area, and increase root length, leaf aging, and brought about different changes at growth, growth stage. Physiological changes include uh, modulation or changes in transpiration, stomatal activity, photosynthesis, osmotic balance, water transmission, and leaf relative water content. Biochemical changes include antioxidant defense system, photosynthetic pigments, accumulation of compatible solutes like proline, glycine, betaine, hormonal contents, and secondary metabolites. Proline, that is alpha amino acids, it helps to maintain the plant's stress water status under drought stress. Osmotic adjustment is carried out via accumulation of certain osmolytes, including proline. Proline, that is considered the key osmoprotectant and compatible solute, is an amino acid that play important role in environmental stresses and also act as an antioxidant. It has been reported that exogenous application of proline through different modes are effective for the induction of stress tolerance by modulation of different physiological and biochemical responses. Manganese, that is an important micronutrient for plant growth and development, and it is it is a metal that is an essential cofactor for the oxygen evolving complex of the photosynthetic machinery, catalyzing the water separating reaction in photosystem growth. Manganese is transported into plants via manganese transporters that are transmembrane proteins. It mainly contributes to a wide range of enzyme catalyzed reactions such as hydrolysis, phosphorylations, and decarboxylation. The deficiency of manganese increases the biological damage by disintegration and destabilizing photosystem to complexes that initiate the production of reactive oxygen species. Rice straw extract, rice that can accumulate about 41.6 silicon per kg dry weight. Silicate minerals accumulate in epidermal cells and form a protective barrier and reduce the water loss during the using the cuticles. These minerals not only strengthen the plant cell wall but also enhance cell wall elasticity through extension growth. Manganese has been reported to enhance plant growth under drought stress by maintaining plant water status, photosynthetic efficiency, and the structure of xylem vessels under high transpiration rates caused by high temperature and moisture. Therefore, many countries utilize the rice straw to enhance the soil nutrient contents. It has been hypothesized that foliar application of proline, manganese, and rice straw extract can increase drought stress tolerance in wheat plants. The main objectives were to check the response of wheat plants to foliar application of proline, manganese, and rice straw extract under drought stress, to investigate the pattern of the changes in terms of growth and physiobiochemical parameters of wheat plants when treated with mixture of proline, manganese, and rice straw extract under drought stress and non-stress conditions. The experiment was conducted in the research area of Government College University, Faisalabad. There were two wheat varieties, and the seeds of these varieties were collected from Ayub Agricultural Research Institute, Faisalabad. And the design of the experiment was a split plot with three replicates of each treatment. There were two water stress levels, normal irrigations and 50% reduced irrigation. There were only two irrigations during the whole life cycle of the crop, and there were 5 levels of foliar applications of manganese, proline, and rice straw extract. For example, there was control without any spray, was simply water spray. The second level was manganese 0.1%, proline 1%, and rice straw extract 10%. And then the fifth level was mixture of manganese, proline, and rice straw extract in 1:1:1 ratio. The data collected includes both attributes like shoot fresh weight, root fresh weight, photosynthetic pigments, oxidative stress markers like malondialdehyde, hydrogen peroxide, enzymatic antioxidants like superoxide dismutase, peroxidase, catalase, and proline. Results obtained showed that shoot fresh weight and root fresh weight significantly decreased under drought stress. However, the applications of proline, manganese, and rice straw extract enhanced the shoot fresh weight and root fresh weight of both wheat varieties NH 617 and MS 2021. Chlorophyll pigments that were chlorophyll A and chlorophyll B significantly decreased under water stress in both wheat varieties. However, the applications of proline 1%, manganese 0.1%, and rice straw extract 10% enhanced the photosynthetic pigments that were chlorophyll A and chlorophyll B in both wheat varieties. Malondialdehyde and hydrogen peroxide contents increased under water stress in both wheat varieties, and this production of MDA and H2O2 was more in MH 2021 as compared to NH 17. However, the foliar applications of manganese, proline, and rice straw extract decreased the production of MDA and H2O2 in both wheat varieties. Activities of superoxide dismutase and catalase increased under drought stress. However, the foliar application of manganese, proline, and rice straw extract further enhanced the production or the activity of peroxide dismutase, catalase, and peroxidase enzymes. EOD activity also enhanced under drought stress in both wheat varieties. The activities of SOD, POD, and catalase was more in ANAJ 17 as compared to MS 2020. Proline significantly increased under drought stress in both wheat varieties. However, the foliar application of manganese, proline, and rice straw extracts further enhanced the production of proline in both wheat varieties. In conclusion, the drought stress significantly decreased the growth biomass attributes of both wheat varieties on R17 and MS 2021. However, the foliar application of proline, manganese, and rice straw extract decreased MDA and H2O2 contents by upregulating the antioxidant defense system and increased the chlorophyll contents, growth, and yield rates under both normal and drought stress conditions. Accumulation of osmolytes that were mainly proline increased activities of antioxidant enzymes. It has been suggested to be an adaptive strategy to combat water stress. The variety-specific response revealed that the NH 17 was excelled the MS 2021 in growth and yield traits under water deficit stress and could be used for cultivation in drought affected areas.